A very good evening to everyone. On behalf of Indian Association of Medical Microbiologists, Karnataka chapter, it's my privilege to welcome you all to this session of the master class. This is the eighth in the series which we have been doing from June 2025. And before we start, a very warm Dasara and Navratri greetings to all of you. And I think fittingly we have the expert from the royal city of Mysore, with us for this session. And before her, Dr. Lochana, who is a postgraduate, and Dr. Mahesh Kumar in Karnataka Medical College and Research Institute, Hubli. She will be presenting, and later on, we'll have the expert session by Dr. Rashmi Mahale from the JSS Academy of Higher Education and Research. Just a brief biodata of Madam Dr. Rashmi. She is currently a professor of microbiology at JSS Medical College, Mysuru. She's also the chairperson of the Hospital Infection Control Committee at the JSS Hospital, and she's also the member of the Antibiotic Stewardship team. Apart from that, she's also the member of the Patient Safety Committee, and she's worked as the head and presently mentor of the Quality Assurance Department of JSS Hospital. Her areas of interest include drug resistance in bacteria and prevention of drug resistance, antibiotic stewardship, hospital infection control, and she has guided many MBBS, MD, PhD, and MSc students, and she has 33 publications in national and international index journals. And one of the other things which I would like to highlight is that she has completed her MBBS and a PhD, both from her alma mater, JSS Academy of Higher Education and Research, and she has done her MD from Kasturba Medical College, Mangaluru. Thank you so much, ma'am, for agreeing to be with us during this session. I also welcome our National President, IAMM, Dr. M.B. Nagmoti, sir, who's been a constant support to all of us for all these sessions. And I sincerely thank Dr. Sudhindra for bringing Dr. Rashmi Madam on board for this session. Thank you so much. I think I'll hand over the forum to Dr. Lochana for her presentation.
Thank you, sir. Sir, can I start?
Yes, please.
Okay, thank you, sir. So, good evening to one and all present.
Sorry.
Sorry, sir.
Can you please put the slides on slideshow?
Yes, sir. I have done.
Okay. I'm not able to use that. The slides are there, but it's not in slideshow. I request others to.
It's not in slideshow.
It's not in slideshow. Probably you can exit from this the slide, and then you can try doing
Yes.
that. Yeah. In the meantime, I would like to introduce Dr. Lochana. She is currently a first-year postgraduate, and it's nice of Dr. Mahesh to encourage his postgraduates to come and present on this platform. So wishing the very best of luck to Dr. Lochana, and I'm sure I mean there'll be a lot of takeaways from this presentation. And Dr. Rashmi Mahale, ma'am, she will be there to take us through the other challenges and the troubleshooting which we have for biofilms. Yes, Dr. Lochana. I mean, it's perfect. Perfect. Please go ahead.
Yes, sir. Thank you, sir. Thank you so much. Good evening to one and all present here. My name is Lochana. I'm first year PG from Karnataka Medical College and Research Institute, Hubli. My mentor is Dr. Mahesh, sir, who's also the head of department in Karnataka Medical College and Research Institute, Hubli. So I'll be presenting on the topic biofilms. So the main areas which I will be concentrating on is the who founded, that is the history. So I'll be talking about the history, the definition with the examples, the basic biofilm structure, the development of a biofilm, its application, the laboratory methods, and how to prevent the infection, biofilm formation. First, Anton von Leeuwenhoek first described about the biofilms in the late 17th century by using his simple microscope and took the biofilm present on a tooth surface. He defined it as a bacterial cluster that aggregated on the surface, forming a polymeric matrix synthesized by them. The biofilms are mainly found on surfaces with high moisture content. Zobel had observed that the number of the bacteria present on the surface was much higher than the surrounding medium. Cherklis studied that the microbial slimes found on the industrial water system were more resistant to basic disinfectants such as chlorine. Costerton explained that the mechanism of how the microbial cells adhere to a surface and survive. Jones used electron microscopy to study the morphology and structure of the biofilm. So now coming to the in the past last 2 decades, the major thrusts which were done in biofilm is that the usage of confocal laser scanning microscope to characterize the biofilm structure, and the usage of higher automated methods such as PCR to find out the gene responsible for biofilm formation. So right now, coming to the definition of a biofilm, it is a three-dimensional structured community of microbial cells which consist of bacteria, fungi, or algae embedded in a self-producing matrix of extracellular polymeric substance that adhere irreversibly to a surface. As I had mentioned, it is a three-dimensional structure that protects the microbial community from biotic and abiotic factors such as predation, toxic substances, and other environmental stress. So biotic factors include means living or once living surfaces, which includes heart valves, teeth, lungs, especially in cystic fibrosis patients, middle ear, skin or mucous membrane, whereas abiotic surfaces means non-living surface, which includes medical devices such as catheters, plumbing pipes, industrial equipment, joints, prosthetic joints, etc. The biofilm is mainly formed by microbial cells during the stress, environmental stress, and it is a type of a defense mechanism developed by certain microorganisms to survive in hostile environments. Viruses do also form biofilm-like structures by utilizing the host cells to replicate. Example being a human T cell leukemia virus type 1. The biofilms not only form on hydrophobic surfaces but also on hydrophilic surfaces. Any surface which has roughness on its surface, biofilms tend to form on such surfaces. So the difference between bacterial and fungal biofilms mainly in the structure of the biofilm. So the bacterial biofilms, they mostly consist of a single species of population. The matrix are mostly composed of polysaccharides. They adhere to medical devices such as catheters and are present on wounds and tissues. The resistance is mainly developed due to quorum sensing, which is a cell-to-cell communication between the microbial cells present in the biofilm. And when observed under the microscope, we can observe that microcolonies in cocci or rods are embedded in a matrix-like structure. Whereas coming to a fungal biofilm, it mainly consists of multicellular hyphae network made of yeast or hyphae. The matrix is mainly composed of beta 1, 3 glucan and mannins. They adhere mainly to mucosal surfaces. The resistance is mainly developed due to efflux pump mechanism and altered sterol synthesis. And under microscopy we get to observe dense hyphal meshwork which are more complex and thicker. They switch between yeast and hyphal form. Now coming to the main common microbial organisms which tend to form biofilms. Under bacteria, we have Staphylococcus aureus, Staphylococcus epidermidis, Streptococcus mutans, Escherichia coli, Vibrio cholerae, Proteus species, Klebsiella species, Pseudomonas aeruginosa, and Pseudomonas fluorescens. Pseudomonas aeruginosa with Burkholderia species are mainly form biofilms in cystic fibrosis patients, and Staphylococcus epidermidis and form biofilm on orthopedic prosthetic devices. And coming to the fungi, one of the most common biofilm forming organism is Candida albicans. Now coming to the structure of the biofilm, so it mainly consists of microbial cells. These microbial cells may include one or more species that coexist together. Next, coming to polysaccharides, this forms the matrix. It gives structure and protects the microbial cells from the environmental hostile environment.
Hello, ma'am.
It also forms the sticky framework for the. It forms the sticky framework. It mainly consists of mannose, galactose. Examples are Pseudomonas aeruginosa has alginate property, whereas Escherichia coli has colonic acid as a polysaccharide. They are produced more when the cells are under stress. Coming to the extracellular proteins, they are present on the external surface of the microbial cells. They form the building block and also helps in the aggregation, attachment, and clustering. Coming to the nucleic acid, that is the extracellular DNA, they are liberated during cell autolysis or during horizontal gene transfer. They are a significant contributor for the formation and expansion of the microbial cells, leading to the formation of biofilms. They are also one of the main reasons for the development of antibiotic resistance. Biofilm formation. So the 5 major steps in biofilm formation are reversible attachment, irreversible attachment, cell proliferation, biofilm maturation, and disintegration. So coming to the irreversible attachment, the microbial cells, few microbial cells attach the surface or a substratum, and they tend to divide over there. At this stage, if any external force is applied, these cells can disseminate. Once they start colonizing, they become irreversible, and later they start multiplying. Once they start to multiply, they start excreting the matrix, which protects them from the external surfaces, also making them irreversible. Once they have matured, they form a complex structure, a microbial cell community. Once this is formed, the microbial cells tend to crowd on the surfaces where there is more water current flowing. So, coming to the example, Pseudomonas has mushroom-like structures, whereas Streptococcus mutans develop tower-like structures. This is another simpler diagram of biofilm formation. So first there is a reversible attachment. Once and then the cells start to develop, multiply. Later it becomes into irreversible attachment. Later the cells start to proliferate more, and during maturation they excrete the matrix. Okay, coming to the application of the biofilm. Biofilms mainly can be if genetically engineered, they can be useful for mankind. But if not, if they mutate, then it becomes a major con. So coming to the applications in public health sector, it can be used in environmental wastewater treatment, as there is presence of organic pollutants in the wastewater. It degrades this substance and lets it the biofilms start uh, uh, breaking down complex compounds into simpler compounds. Next is bioremediation of pollutants like hydrocarbons and heavy metals. They break down harmful substances into less toxic or non-toxic substances. They also have the power to efficiently clean up the environment in substantial and cost effective manner. Coming to the agricultural biofertilizers, they are genetically engineered biofilms can be introduced into the soil, making it more nutritious, and can also be coated on plant roots for the better absorption of nutrients from the water, and also used in biofueling. Coming to the major disadvantages in public health sector, we have it contaminates food products and food processing industries, leading to uh, not being cost effective, and development of biofilms in irrigation lines can clog the flow. Uh, Coming to the healthcare setup, the major one most common uh, harmful effects. So chronic biofilms form a chronic and biofilms prevent in faster healing, leading to a chronic and recurrent infection. Development of biofilms on indwelling medical devices such as catheters are such a nuisance. Hence, the patients have to be started on antibiotic therapy. Spreading of antimicrobial resistance due to quorum sensing and horizontal gene transfer, and there are many mutations occurring during the cell division. It leads to more antimicrobial resistance development. In hospital outbreak reservoirs, the biofilms tend to grow in disinfectants, making it tough to use disinfectants to clean the to get rid of the biofilms, and causes diagnostic challenges. So collecting biofilms from indwelling medical devices is little tough. From collection of samples to processing the biofilms for diagnosis, though if present in less quantity, it leads to a false result. Okay, so the common organisms which are associated with which are isolated from certain devices are uh, artificial hip processes. The common organisms which form biofilms are Staphylococcus aureus and Staphylococcus other than Staphylococcus aureus. In central venous catheter, Klebsiella pneumoniae, Staphylococcus aureus, Pseudomonas aeruginosa, Candida species are were isolated, and in most of the organisms are mainly isolated from urinary catheters. Next, coming to the laboratory diagnosis. So, laboratory diagnosis for biofilms can be divided into qualitative and quantitative method. So, quantitative method includes differ helps us to identify how much biofilm is present. It is a more detailed method where it is interpreted either as positive or negative, or strong or weak. Examples include tube method and Congo red agar method. Whereas qualitative methods, it helps us to identify whether the biofilm is present or not. It is used for quicker screening, and it is interpreted in numerical values. Example, tissue culture plate method and crystal violet assay. So coming to the laboratory diagnosis, the first main one which we use to identify the biofilms, or to talk about the morphology, structure, topography about my biofilm, we use electron microscopy. Under electron microscopy, we have two types, which is scanning and transmission microscopy. The electron microscopy, scanning electron microscopy is much preferred, as it helps us to identify the three-dimensional structure of the biofilm. It also gives us a better, higher magnification, and there is no need of sample preparation. Whereas in transmission, only a two-dimensional structure can be observed, and the sample has to be prepared, such as dehydrating the sample. Since biofilm is mainly made up of a matrix which the major content being water, it is tough to observe. So the better microscope right now used to study the morphology of biofilm is confocal microscope. The limitations for both these microscope are it's mainly used for research purpose and it is expensive. So this is a morphology structure of a biofilm under a scanning electron microscope. Coming to the Congo red agar method. So here the Congo red dye solution is autoclaved separately and added to the brain fusion agar, which has sucrose at 55 degrees Celsius. The plate is inoculated with a test isolate at 37 degrees Celsius for 24 to 48 hours aerobically. After 24 hours, the results which we interpret is black dry crystalline colonies indicate they are strong biofilm producers. Just black colonies indicate moderate biofilm producers. Weak biofilm producer show up as a dark pink colony, and non-biofilm producers produce dry red colonies. So this is the image where black dry crystalline colonies indicate strong or moderate biofilm producer, and dark red as weak biofilm producers, and dark pink as a weak biofilm producer, and red as non-biofilm producer. Now coming to the tube method, 10 ml of trypticase soy broth with 2% of glucose is inoculated with a loop full of the test isolate overnight. It is incubated at the 37 degrees Celsius for 48 hours. Then the tubes are decanted, and excess of the tube is poured out if it consists of excess broth, and the tubes are washed with phosphate buffer saline. Later, the tubes are dried and then stained with 0.1% saline violet iron for around 13 minutes. Later, the excess stain is decanted with the deiodinized water, and the tubes are dried in inverted position. So to interpret a tube method, we see if there is a formation of a visible film in the walls and the bottom of the tube. If in case of presence of a ring formation, it is considered as a negative result. And this tube test is graded as 0, which means absent, 1 showing weak, 2 being moderate, and 3 being a strong biofilm producer. Coming to the tissue culture plate method, which is the gold standard method. So the suspension of the test isolate, the test isolate is suspended in a Muller-Hunton broth. The broth is then transferred to a 96 well microtiter plate, which is incubated at 37 degrees Celsius for 24 hours. The wells are then washed with buffer or sterile distilled water so as to remove the planktonic cells. Later, it is stained with 0.1% crystal violet, which is left at room temperature for 30 minutes. Again, it is washed out with the buffer solution or sterile distilled water. Late and It is washed minimum 3 to 5 times. Later, 95% 90 to 95% ethanol solution is added. And is washed out. Later, the spectrophoton is used to find the optical density for dye of the ethanol dye suspension. So this indicates the tissue culture plate method. Coming to the limitations of the tests, there could be observer bias. It does not help us to differentiate between live and dead cells, seldom used except for research purpose, and hence automated methods are most preferred for faster and better diagnosis. So coming to the automated methods, first is polymerase chain reaction, which is highly specific and sensitive method for detection. It detects the genes that encode for the matrix formation. It is used for detecting the biofilms forming pathogen from the clinical samples. Examples: real-time PCR, multiplex PCR, and reverse transcriptase PCR. So the common genes which are involved in biofilm for production is each organism has a different gene. So Pseudomonas has the gene ALGA called as alginate biosynthesis gene. Streptococcus mutans has the gene MUFC regulator. Staphylococcus biofilms have the gene ICA genes, and Candida albicans has GSC1 and MNN1 gene. Next, coming to Malditov MS, that is matrix assisted laser desorption, ionization, time of flight, mass spectrometry. It compares the microbial protein profile from the database of reference mass spectra for identifying the genera, species, and subspecies. The identification is performed less than 1 minute. The disadvantage being when the same organism was repeated, it showed different results. Next, coming to omics analysis, it analyzes the entire microbiome of the pathogen. It also helps to analyze the gene expression, protein, and the metabolites produced by the microbial cell. The disadvantage being we cannot compare one data with another, and the analysis is done from a single cell from the entire population, making the subpopulation neglected. So coming to the prevention of biofilm, especially in the healthcare setup, coating the surfaces with antimicrobial agents. The prosthetic devices, if coated with antimicrobial agents or using of anti-adhesive surfaces, is much helpful to prevent the development of biofilms. Enhance cleaning and disinfecting the healthcare setup regularly. And say per se, if the patient has developed biofilm on temporary devices such as catheters or central venous catheters, urinary catheters, we are advised to immediately remove the device and to start on intense antibiotic therapy. And dual antibiotic agents are always preferred compared to single antimicrobial agents. Thank you.
Thank you so much, Dr. Lochana. I mean, it was a wonderful presentation. So
Thank you, sir.
keep it up, and I'm sure you have laid the foundation for the expert talk by Dr. Rashmi, ma'am. I mean, for those uninitiated, Dr. Rashmi has worked a lot on biofilms and antimicrobial resistance being one of her core areas of interest. I think she will be the right person to talk about this and give us her insights about the biofilms and how to deal with it. Over to Madam.
Thank you, sir. So first of all, I congratulate Dr. Lochana for her good presentation. But few of the things which I would like to emphasize on is the clinical burden of infections which we are having due to biofilm producers. Around 80% of all infections are due to biofilm producers, and among them, 62% of the nosocomial infections are due to biofilm producers. So that is the burden of the infections which we are getting due to biofilm producers. And as Dr. Lochana said, the treatment of these biofilm producers is very difficult because they result in recurrent infections. So now, as she said, they can be defined as sessile community of bacteria which are embedded in a EPS matrix. So those are the biofilms. So now these biofilms, it is just not sufficient that it is enough that we identify the organisms are biofilm producers, but we also need to know how we have to get rid of these biofilms, not just by prevention, but once the patient is infected, we also need to know that there is something called as minimum biofilm eradication concentration. So now, though this is a tedious procedure, but this can be done when you encounter recurrent infections due to biofilm producer, and we know that it is because of a biofilm producers. So now this MBEC or minimal biofilm eradication concentration technique, detection technique, is quite cumbersome because what we do for MIC, the same thing we need to do in MBEC, but for each concentration of the antibiotic which we use, we have to use a different PCR peg on which a biofilm has been formed. And then we have to know whether the biofilm has been eradicated at that particular concentration or not. So 100% reduction in the growth of organism at a particular concentration of antibiotic, then that concentration we call as minimal biofilm eradication concentration. And one more thing she told, I think she missed to say, was how these biofilm organisms in biofilm are resistant to the antibiotics or even the host immune responses. So as students, so whenever you have a question which is asked on biofilm, it is important that you tell or you write about how this resistant mechanism is induced in these biofilm producers, which is not seen in planktonic bacteria. So now, can anybody tell how do they develop resistance? Okay, if nobody is going for that question. So now this biofilm, when the bacteria are embedded in the matrix, so their level of multiplication is little less. So, and as we know, the antibiotics will act on actively multiplying organisms. So this will tell us why they are more resistant to the antibiotics. And also, when they are embedded in a matrix, the host defense mechanisms also do not work. So that is the reason that infections are more resistant to either the antibiotics or even the host immune responses. So next, another thing which I wanted to tell was she also mentioned that they grow in disinfectants. They do not grow in not all the biofilm producers grow in disinfectants. They do not grow in disinfectants. Some of them might grow in disinfectants, but not all the biofilm producers grow on disinfectants. They are usually seen on hospital environmental surfaces. So if you do a surveillance swabbing of the hospital environment, you might isolate organisms which are biofilm producers. Next, I wanted to say one more thing is about quorum sensing. So when you're talking about biofilm, you also need to know about quorum sensing. So quorum sensing is nothing but a communication between the bacteria. So when the bacteria are within a mass, they produce certain substances which are called as autoinducers. So when the concentration of this autoinducer is more, so then the organisms will genetic expression of the organism is going to be changed, and they have the capacities to produce more virulence factor, more antibiotic resistance. All these are going to be more in case of when you see the autoinducers which are being produced. Okay, and one more thing which I want to put into this thing is, she has told about the 3 methods of detection of biofilm. So now she spoke about the Congo red agar and then the microtiter plate. So these 2 are almost good enough for us to do it in the clinical laboratory. So the Congo red agar is easy to do it, and also to compliance, it's very easy to do it. So we can introduce that in the normal day-to-day laboratory technique, but it has to be asked as a different request. So now when I did my work, I saw that 63% of my isolates were positive when I did when I tested them to be whether they were biofilm producers or not by the Congo red method, whereas 72% of them we observed when we did in microtiter plate. However, that tube method, it's not as this thing, because it is subjective. So each person might see the rim, some might not see the rim. So this is not going to, it was not standard. I felt it was not as standardized way of detecting the biofilm production. Now, next she said, and one more thing what she missed when she said about this thing, what is that? Detection by microtiter plate. She just said that it can be read in the spectrophotometer, but it has to be at 570 wavelength. Only then you can do it. Otherwise, it's very difficult. So we had to do it multiple times when I did that. It has to be read only at that wavelength. Only then we'll get the correct measurements. Otherwise, it's not possible. And each time when we do the test, you have to have a control. So it is not just the positive control and negative control what you have to include if at all you are planning to do any project work on biofilm. You also have to inoculate plain media, and then you have to stain it also with your Congo red, with methylene blue, so that you can deduct that OD value from your other OD value which you have got from the organism where you have inoculated the organism. So that is going to subtract, you need to subtract that OD value from that of the ones which you get it from, with the help of the organisms. So that is one thing which you need to remember when you are trying to detect the interpret your this thing. And lastly, the PCR method, what she said, you have various methods. So first you should know what are the inducers, what are the genes which help in biofilm production. So one is your adhesins. The first thing which is required for a biofilm production is adhesins. So all the genes what she mentioned were adhesins, the genes which are coding for adhesins. So next one is the regulatory genes. So these are also included. This is what these were all. I have also tested for these. I have compared the biofilm production with the phenotypic method as well as the genes which were coding for biofilm production. So that was my thesis topic, which I did quite some time back. So these are the things which you need to remember when you want to work on biofilms. So this is what I had to tell about this presentation of detection of biofilm methods. And I think now the forum can be open for any questions and answers.
Thank you so much, ma'am, for your valuable insights. And I think we'll open up the session for any queries. And I think madam will be happy to answer those queries. So, I mean, if there are any queries, please raise your hand so that or people or participants can even post the queries in the chat box.
Might be next application might be you'll have to have an anti-auto inducers to prevent the development of biofilms. That's what is the next generation thought.
I'm Dr. Navina, Dr. Satyanarayan. Can you hear me?
Yes, ma'am. Yes, ma'am. Please go ahead.
Yeah, this is the first class I'm attending. I just had to see this in KC. It's quite interesting, and I would continue to attend these. These are mainly for the PGs, is it?
No, ma'am, it's open for
Masterclass
it's open for
open for.
everyone. It's
Yeah,
open for everyone.
it's quite an interesting topic today, biofilm. I have one question. In the routine lab, diagnostic, how is it going to, though we know that this, some of the catheters, for example, central line could be, when there is a CRBSI, probably we think it is causing infection and we tend to remove the catheter. So how does this routine detection of this biofilm formation help in the management of the patients?
The biofilm forming organisms might not respond to the normal MIC value which
Normal
we give.
MIC. Okay.
Okay, so then we'll have to, that's when I said your minimum biofilm eradication concentration might have to be included. But as I said, it's little cumbersome to do it, but it can be done. When you have a recurrent, persistent infection or something, you can attempt on it.
Okay. And is it valid to routinely test for this biofilm formation in some of the isolates? For example, when there is a case of VAP or CRBSI or COTI?
You can attempt to do it, but it won't be requested for, and you will not know what to do later on, because even if you know
Yeah.
it is a biofilm producer and you're not going to do a minimum biofilm inhibiting concentration, then it is not going to be of much help. Only thing what you can think of is prevention of spread of such organism to the other patients. You have to take contact precautions so that you do not spread the such infections organisms to others.
Okay. Thank you, doctor. Thank you.
Thank you, ma'am. Ma'am, if I can be the messenger, there are two queries in the chat box.
Yes.
The first one is by Dr. Sheila Joshi. She's asked for explanation on formation of biofilm.
Lochana, do you want to take that question?
Sure, ma'am.
Yeah.
So the biofilm, there are five main steps. First is irreversible attachment to a substratum, then reversible attachment to a substratum, then irreversible attachment to a substratum, then the maturation of the biofilm, then disintegration. So coming to the first step, which is reversible attachment, small certain microbial cells attach to the substratum with the help of pili fimbriae or a flagella, and they start to divide on the surface. At this stage, they are sensitive to antimicrobial agents as well, and they can also, if disrupted, can move to a different area. As the cells start to divide, they get irreversibly attached to the substratum. Once the cells start dividing, they start maturing. During maturation, the extracellular matrix is produced, making the microbial cells, protecting the microbial cells from the environmental stress. And later on, they disintegrate and move on to the other places.
Thanks, Dr. Lochana. If I can move to the next question, ma'am. This is by Anuradha, ma'am. She's asked you to kindly highlight on the collection of samples for detection of biofilms from devices.
Lochana?
Yes, ma'am.
Yeah, how do you collect samples?
So always secretion, the slimy part present on the catheter is much preferred instead of sending the cut portion of the catheter.
No, is it accepted? Catheter tips, are there? Which
No,
catheter tip
no,
is
ma'am.
accepted?
Catheter tips are not accepted. The slime present, or the secretion present on the catheter should be accepted, can be accepted.
You're not going to get the slime. How are you going to collect the slime?
Swab, ma'am.
Hmm?
Swab.
Not swab. Anybody else? So now if it is a urinary tract infection, urine itself might be sufficient, or if at all you're going to have this thing, you can flush out the catheter and take the sample from there. Is it okay, madam?
Anuradha, ma'am, you want to come in? Yeah.
Hello, good evening.
Good evening, madam.
ಕೇಳಿಸ್ತದಾ?
ಕೇಳುತ್ತೆ ma'am.
Am I audible?
Yes, yes.
Okay, okay. No, because there is a lot of discussion about collection of sample for detection of biofilms. Many people say it is not
Madam,
required to take from the devices and all.
madam, video on my madam. We want to see you.
Hey, beda, beda. Thank you.
Yeah, we want
No.
to. We happy to see you, madam.
So that is why I raised that question, like, because many PGs are attending this session. So routinely we don't look for it. Like, how do you like to tell them to do that? Like, of course, generally we flush this tubes, cannula, what we get. But is it fine, or do we have to take from? Because if you take from outside, they say it is colonizers mainly. It may not be a pathogen.
I think we can take the flush.
Flush is
Flushing
better.
out, it is better, Sam.
Flush is better. Thank you.
I have to acknowledge this. Madam was my guide in the PhD. She was my co-guide for my PhD topic.
Thank you.
That's nice. So, any other queries?
I have another question, Dr. Satyanarayan.
Yes, ma'am,
What
please, ma'am.
about we are reusing this cardiac catheters, right, for the patients, and whether the sterilization reaches this biofilm, all this ATO and plasma?
I think Dr. Rashmi will be happy to answer that, ma'am.
So even I don't have much of an experience with the sterilization because I have never evaluated anything of that. But it is a topic which thought-provoking topic which we can try.
Since I come from a
cardiac.
Can I tell? I
Yes,
think
madam.
you can clean it first with the water, hot water, and proteinaceous material has to be removed. Then you sterilize it by ETO. I think that will work. Low temperature.
Enzymatic.
All the proteins that are deposited on the cannula can be washed thoroughly. Then you sterilize it. I think that
all this pre-treatment happens.
Enzymatic digestion happens.
Yeah, pre-treatment and all that happens routinely, but still, whether if some organic matter is there, whether this biofilm is there, whether the sterilization will be effective on the biofilm. This is my question.
I think it should, but we can
be.
Yeah, thank you.
Yeah. Next, we have one query from Dr. Arvind Singh as well. He's asked whether it will be legitimate if we suggest clinicians for requesting biofilm diagnosis in case of patients with implants if a biofilm producing organism is suspected.
Whether legitimate biofilm organism, you have already detected that it is a biofilm causing infection.
I think his query is whether you can specifically ask for biofilm diagnosis, ma'am, in patients who have implants.
That I think it is left to us whether if we are getting a sample recurrently from a single patient because of recurrent infections, with your interest you can do and try to find out whether the patient is having a biofilm infection. Otherwise, I don't think in no hospital you have a separate request for it. Right now, we do not have it.
Absolutely. Yeah, I think that answers the query. Next, we have a query from Dr. Sudhindra. He's asked for the role of antibiotic flush therapy for biofilms.
Antibiotic flush therapy, it does, but I do not know the concentration of antibiotic which is present in the flush might not be effective to the biofilm which is already formed in the medical device which is used. So I feel it might not, it might be a preventive aspect, it might help, but not for curing.
Correct. Thank you. I don't see any other queries as of now. I think you answered most of the queries. So if there are any queries, please feel free to share. I think Madam will be happy to answer them. But as of now, I don't see any other query, ma'am. So I'm sure, I mean, the session has been helpful for most of us, and not most of us, it's for all of us. And I think Madam has clarified on the most important things related to biofilms and how to tackle them, how to address the issue. So thank you so much, ma'am, for your presence and your valuable insights. Now, just taking it further, we have one more query, ma'am. Can you explain how to flush whole device should be taken out? I'm not sure that I have got the question right. Can you explain how to flush whole? I think the query second part.
Sample collection, probably he's trying to talk about how
Probably,
to take
probably,
the flush
yes.
sample. So
Probably.
if you get the tip or a catheter segment, probably you have to, with using sterile saline and syringe, you have to flush out. And whatever you're getting, you have to take that as a sample.
Dr. Arpita, I think that's answered your query.
Mahesh, sir. Dr. Mahesh.
Hello, Sudhindra. Yeah.
Sir,
Hello.
you would like to hear from you, sir.
Nothing. Hello. Yes. Tell me, Sudhindra. Dr. Sudhindra.
This is first of IAMM-PG Masterclass. This is the first of its kind. Different.
It was.
Thanks a lot for Mahesh sir for the initiative. We'd like really
I
congratulate you
need
for
to thank the. I need to
initiative
thank. So the other way around, I need to thank the IAMM-KC office bearer for giving. Hello. Hello. Hello.
This concept is your brain side, sir. This is your brain side. Getting the PGs into the.
The whole idea started to improve the PG. Hello.
Yeah, we can hear both of you. Please go ahead.
Yeah. Sudhindra, other way around, I need to thank the IAMM-KC office bearer for giving platform to the PGs. The whole idea of starting this means when the discussion started is to improve the quality of PG teaching, right, Dr. Satya?
Yes, sir.
Yeah. Yes, so in that way, I just suggested this way. That's it. Nothing great of anything. Thank you, Dr. Rashmi madam, for giving your insight and your sharing your experience. Really, it is very helpful for those who have knowledge only of the book. You have a knowledge of hands-on knowledge. Thank you, madam.
Thank you, sir, for giving me this opportunity.
Yeah, this as was just detailed by Dr. Sudhindra just a few minutes back. This was like a hybrid model where we had a PG presentation followed by an expert talk. And Madam has addressed most of our queries. So thanks to Dr. Lochana and Rashmi ma'am for your presentations and your insights. On behalf of the association, myself, I'm Dr. Satyanarayan. I'm from Department of Microbiology from Bangalore Medical College and Research Institute. I thank all of you for your valuable time and for being present for the session. And have happy Dasara, happy Navratri to all of you. Thank you so much.
Thank you, Dr. Satya, and thank you. Thank you, Satya. Thank you, Sudhindra. And also thank you, Rashmi madam.
Thank you, sir. Thank you so much.
And all the people who listened. Thank you all.
Congratulations to Dr. Lochana.
Thank you, sir. Thank you so much for giving me this opportunity to perform on this stage, sir. And I thank to Dr. Mahesh, sir, for helping me in this presentation. Thank you, Rashmi, ma'am, for giving your insights about biofilm. And thank you, sir, for helping me out and giving me this stage to perform. Thank you, sir.
Thank you.
Thank you.
I'll stop the meeting.
Yes.
And the presentation, the recording of the session will be available. I'll be sharing the link with most of the groups. I think you can pass it on from there. Thank you so much.
Thank you, sir.
Good. Thank you.